In other cases, we found that they used a rather loose criterion for determining salinity tolerance. They said if a fish has salinity tolerance to 14 parts per thousand brackish water, then it could survive and get into the Great Lakes. In fact, we know right here in the Montreal area, either US or Canadian inspectors board every vessel and they check to see if the water is 30 parts per thousand, which indicates ocean water. If it's not 30 parts per thousand, it is illegal for them to discharge it into the Great Lakes, and it gets checked as they're leaving. So the ships comply with that rule. Consequently, if you're going to invade the Great Lakes, you need to have ships coming from ports where the fish occur in Europe, number one. And number two, those fish must be able to tolerate when the ship captain is going to turn on the ballast pumps on the ocean and flood them with salt water. So what we break this all down to, the so-called 10 high-risk species, we think there's actually one, the big-scale sand smelt, that still constitutes a risk. There's another one that comes out in the middle because there are about 70 ships over a 2-year period that came from ports where this fish occurs, and it can tolerate salt water. However, when you go back and look at the literature, there are no reports of this species being an invader in any system or causing problems in any systems in Europe. So even if it did get into the Great Lakes, of course it could act differently here than it does in Europe. But we think precedents in other systems are very telling. And in this case, there are no precedents to indicate that that's a problematic species. So really, we reduce this from 10 down to 1 species. We would love to see some of our colleagues in terrestrial, who work in terrestrial environments, apply the same type of criteria, looking at colonization and propagule pressure differences in communities over time, or in pathways. Some of these things like horticultural soil that's moved around, hay and straw, they can move lots and lots of species from one location to another. And of course the wood dunnage that I mentioned. Boy, can you read that at all? You can't. This is from a previous study that we conducted, and then we reassessed some of the data in 2011. And what we were doing here is the ships were starting in a freshwater environment, and then half the ballast tanks would be exchanged on the open ocean. The other half would not be exchanged until it got to the destination. We would monitor the populations when the water was first put into the tank, and we would monitor the water just before it was discharged at the end of the trial. And what you need to know is this column right here, the efficiency of scrubbing out freshwater species if you do ballast water exchange before coming into the Great Lakes. We have values that range, this is Brian Leung giving us 4 decimal places, 3 decimal places, 99.988% or higher. So this is why we think, at least for invertebrates, that ballast water exchange is very effective for vessels coming into the Great Lakes. Despite that, the UN agency, the IMO, is going to switch over from a mandatory ballast water exchange policy to one of mandatory ballast water treatment. And when this treatment is done, they're going to apply what's called a performance standard. So in order for a system to be approved for treating ballast water, it must be able to achieve these levels for each of these indicator organisms. So for zooplankton-sized organisms, you must have fewer than 10 viable organisms in a cubic meter of water. For someone who studies plankton, if I go out in the lake during summer, I might find 50 of these size organisms in 1 liter of water, or 50,000 in a cubic meter of water. So this standard is very robust for zooplankton-sized organisms. The standard for phytoplankton-sized organisms is much less robust, and then we have a number of indicator organisms for bacteria, E. coli and Enterococcus. So, and I mentioned here, if you look at, in order for this treaty to be ratified, it needs a minimum of 30 countries. We're at 46 right now, so that part is passed. And we need a minimum of 35% of global shipping tonnage carried on the world's oceans. We're at almost 33% right now. Singapore's about to sign on. Singapore's a huge shipper. Once they sign on, the treaty's international treaty that will apply worldwide will be ratified, and then this new water treatment standard will come into effect 1 year later, okay? So what we wanted to do, and we did this study in conjunction with a company headquartered here in Montreal, FedNav, we wanted to see, after they bring in the ballast water treatment, this performance standard, is there any utility in still requiring ships to flush water through their tanks as they come across the ocean? They don't have to stop. They do it while they're running. And so if there's no cost to them, why not use it as a fail-safe to make sure that if something happened with the pumps, you have crews on board the ships. They may see a red light, but they don't know what it means. And so this would provide us with a safeguard that it's unlikely that the ships could still carry in species because we still have the ballast water exchange to fall back on. So statistically, what we then did was test a number of different models where there's no interaction between or no benefit from adding ballast water exchange to ballast water treatment. What we were looking for is this one right here, where combining the two together yields something greater than the individual effects by themselves. Not quite as good, but still good, would be an additive response where it's simply one added to the other, which would mean in terms of plankton that the plankton abundances go lower and lower, propagule pressure. And then there are two antagonistic ones where the effect is less than the additive effect of the two individual treatments. So we conducted a study on a ship that's now been sold called the Federal Venture. And the main participants in this are Esteban Peluche, who's now a scientist with the government down in Argentina, and Marco Hernandez, who's still a PhD student in my lab. These are our 4 treatments. The beauty of these ships is that we have lots of ballast tanks that we can use for our experiments. We have to be careful that we don't mix treatments with or water lines that have contained chlorine with water that is not supposed to contain chlorine. And so what we had to do here, we have 2 ballast water pumps serving this vessel. The port side one, which is receiving chlorine whenever the pump is turned on, and the starboard side, which is not receiving chlorine. And we have controls, chlorine only, ballast water plus chlorine, or just ballast water exchange. Very hard to see. So most of the trips began in the upper St. Lawrence near Port Alfred. They're loading freshwater ballast. We would collect samples as the water's being loaded. When it's out on the open ocean, they're going to conduct ballast water exchange for the tanks that require it. And some of those tanks also require chlorination, so they would be rechlorinated a second time. And then before we get down to Brazil, we have to discharge all the water because they don't want raw ballast water and they don't want chlorinated water in their ports. So we would stop somewhere along this line. And we did 5 of these trials. These are somewhat complicated graphs showing the abundance of our different indicator organisms. 5 trials. The one I want you to pay attention to is in the lower right, where we're looking at the average of our different trials. These are for Enterococcus bacteria, the abundance of these in the different tanks at the outset of the experiment and at the end of the experiment. The red dotted line indicates the new performance standard that's going to come into effect when the ballast water treatment is ratified. You can see here, this is for a control tank. Clearly very high Enterococcus levels in the water that was loaded in Quebec. It falls a little bit, but it's still well above the international permissible level. Chlorination brought it down to a level below, not surprisingly, below the IMO safeguard level. Ballast water exchange was totally ineffective at preventing or at reducing the Enterococcus numbers. And then we have this one down here, which is near 0. Statistically, if we look at that result, the main thing we're interested in here is this last row, where we have an interaction between ballast water exchange and chlorination of the water. We have a negative effect here, and overall there's a synergistic benefit to adding these 2 treatments together. We do the same thing for E. coli, essentially the same results. And again, we have a synergistic effect, reduced populations of both of the bacterial indicators. The third indicator, the Vibrio cholera, we were monitoring for, but we never found any in any of our samples. This is for the phytoplankton size groups, and here you can see that the effect is not quite as clear. A large reduction for both chlorine and chlorine plus ballast water exchange. Again, ballast water exchange by itself is ineffective at getting us where we want to go. If we look at statistically what happens here, there is an antagonistic effect. So overall, the effect was less than an additive one. But it's important to note that in all cases we found that the combined or hybrid treatment always had fewer organisms left than any of the other treatments in all trials. And the last one is for the zooplankton-sized organisms. The arrows simply indicate that there are organisms there. You just can't see them on this graph. And you can see that there's only 4 trials for the zooplankton. FedEx destroyed our first set of samples when we trusted them with them. But overall, you can see that here's our control, chlorination only, ballast water exchange only, and the 2 treatments together. It looks as though we're not accomplishing what we need to get below this IMO permissible limit. However, it's entirely due to one of the 4 replicates. This replicate here, number 2, log axis, relatively high concentration, about 80 and 70 individuals per cubic meter of water, 7 times greater than what's allowable. The other ones were all very low, but when you take the average of those, we were still in excess. The important thing to note, though, as I just mentioned, we tended to see that this hybrid treatment of the 2 combined together appears to be more protective in dropping the propagule pressure down. And in this case, the only significant effect was that due to chlorine. So in conclusion then, the abundances of all the target groups were always lowest when the hybrid treatment was used. Most hybrid treatment replicates were below the permissible IMO D2 standard. These treatments, as I mentioned, they're not going to go out and sample ballast water on the ships as they come in. What they're going to do is companies are going to put their equipment forward in a standard testing environment. The equipment will be challenged, and if it passes the challenge, it can then be put on board any vessel, and it never gets checked again, unless they may have periodic checking to see if the filters are clean and things like that. So we think that you could have undetected failures, and for the simple reason that getting that propagule pressure down as low as you can is going to yield a benefit in that we're going to have fewer expected invasions. We have argued to the federal government, to Transport Canada, that they really should require the international ships coming into the Great Lakes to continue pumping salt water through, even though they have a treatment system on board. Finally, I'd like to thank our different funding agencies. I forgot the CRC program on here. But without all of these different agencies, none of us here in Canada would be able to do our research. We always want more money, and we try and get it for you, but it's not always easy. And with that, I think I'll stop. Okay? Any questions?